Well, before I introduce our speaker, I wanted to give Wendy Stott an opportunity to advertise the next Coragonid Symposium that she has been leading the charge and for organizing. Wendy, would you like to share more information about this with our group?
Sure thing, Bo. Thanks for the opportunity to advertise this. So, like Bo said, we got a group together who is leading the charge on organizing the next Coragonid Symposium, and the more formal name is up there for you to look at. So, as a lot of you know, a lot of you may be familiar with this meeting, but for those who don't, this conference is very international. It's been held since the late 60s every 3 years usually, and usually in the for the past couple of decades now, it's been 2 years in Europe and then one in North America. So the last time it was in North America, it was in Bayfield, Wisconsin, which a lot of you may remember in 2017. The last time it was in Canada was in 2018 in Winnipeg. We're still working on the agenda, but this meeting is kind of cool that it's one session. So everybody's together for the entire meeting. And we usually have several themes that may or may not be familiar to everybody, but we'll have all the usual themes: biology, life history, population dynamics of crigonines, talks on management and assessment, disease, aquaculture, and of course genetics. On these interesting fish. So this year it will be held in Port Elgin in Ontario, and will be hosted by the Saugeen Ojibwe Nation. And this slide just shows you the group that have come together to help organize. So we've got representatives from Canadian and American management agencies and a good group of people that have experience with this meeting. And then we're still sort of underway generating support. So far we've gotten great support from Bruce Power Corporation. So just a little bit more about where. So we've got some dates set, September 20th to 25th, 2026. So mark that on your calendar. And as I said before, in Port Elgin, Ontario. And this is a scenic beach town on the shores of Lake Huron. For folks that work on Lake Huron, it's kind of almost smack dab in between ports at Tobermory and Goderich. It's kind of a tourist town. It's in that stretch of Saugeen Shores with people, sort of the more famous beaches, Sauve Beach, but this is also a nice little beach town with lots of sort of summer activities, even though it's more of a four-season town. Used to go here as a kid all the time for day trips. So it's about 3 hours from Lester Pearson Airport, and you can get there via bus, train, car. So lots of ways to get there. Um, the we've acquired and have secured lodging at a conference center, the Unifor Education Center. And so the meeting will be held there, and we also secured lodging there. So this site is really nice. It's right on Lake Huron. Has These pictures kind of show them the variety of the meeting halls, the cafeteria on the right, an overview of the center so you can see the way it's laid out. There's lots of walking paths, nice forests, it's sort of forested, but also on the lake, and then lots of places for activities in the evening. And in the center is sort of an idea of the lodging. So we have acquired on-site lodging, but people also have the option to stay in town in Port Elgin. It has a selection of hotels and bread and breakfast. It's about a 8 to 10 minute drive from the conference center into town. What else? So just the final slide is just some more pictures of the conference center for you to see. So just kind of keep your ears tuned. Like I said, we've got the site secured. We got a date. Our next thing is to try and get the website up and running, and we've just started working on the program. So we're kind of excited. We're going to try and do some unique things here this year, in collaboration with the Saugeen Nation. And so I hope to see you there. Thanks,
Thank you so
Bo.
much, Wendy. Yeah, that's great. September 20 to 25, 2026. So not too early to book it on your calendars. And Wendy has done the lion's share of work in organizing this team. So thanks for her work on this. All right. So it's my pleasure to introduce our speaker today, Jared Hummela. Jared got his bachelor's in science from fisheries and wildlife at Michigan State University. He then went to Grand Valley State to get his master's in biology with an emphasis in aquatic sciences. He went out to University of Maine in Orono in 2018, where he got his PhD in ecology and environmental sciences in 2018. He came back to Michigan State to do a 2-year postdoc, NSF postdoc, and then became the assistant unit leader with USGS, the Wisconsin Cooperative Fishery Research Unit in 2021. His research program addresses fishery partner needs through genomic, bioinformatic, and statistical tools. And we'll see an example of that today when he shares with us the recent work he and his lab have done on Lake Whitefish. You've heard me say Michigan State twice already in his background, and starting January 1st, Jared will be moving his program to Fisheries and Wildlife at MSU. We'll be working with members of the Michigan DNR Fisheries Division using genetic tools to address their research needs. So congratulations on the new job. Jared, let's see, I think you should be able to share your slides. He will correct the title of his talk here. We had a small typo there. His talk is about 45 minutes, and he's agreed graciously to stay on. And hopefully many of you will have questions and we can have a nice discussion afterwards. So thanks again for doing this, Jared. Thank you.
Yeah, thanks, Boyd. Appreciate the introduction and also the invitation to speak today. I've sat in many of these seminars, and it's great to have a chance to share some of the work my lab's been doing with you all as well. So, like Bo mentioned, had kind of a hiccup in our titling here. So I apologize to any nervous system aficionados that joined today. We won't be talking about anything neural, but instead neutral and adaptive structure of Great Lakes Lake Whitefish. And I plan to, I set this talk up to go over some of the foundational work that we've done to establish a knowledge base on this topic, and then some of the applications that have sprouted from it since. So while my name's the one on the title slide here, it's just because I can't fit the names of many other people that have made the work possible that I'm going to be talking about today. I've had collaborators that have laid the groundwork before I got involved in working on the study system, have assisted all throughout, and continue to build on the science I'll be sharing here. So I just want to acknowledge all these great folks and others that have helped make this work possible. And of course, any errors or omissions in anything I share today are mine alone, including typos on the title of the talk itself. So I know I'm largely preaching to the choir here with this group of folks, but Lake Michigan's Lake Whitefish, which a lot of my talks will be focused on Lake Michigan, they're really important for a variety of reasons. Economically, they're the largest commercial fishery in the Great Lakes and about the only one in Lake Michigan itself. Culturally, it's an extremely important resource, both in indigenous traditions as well as contemporary cultures throughout the region. And as a native species and a key part of the food web, it's also an ecologically very important species. As I mentioned, it's a commercial fishery, so there's harvest that goes on in Lake Michigan and elsewhere throughout the Great Lakes. And harvest is allocated through geographically defined management zones, which also serve as a spatial unit to be able to perform stock assessments, to be able to keep an eye on how well the population's doing out there. And through time, the recruitment of Lake Whitefish and Lake Michigan elsewhere has been really volatile. There's been periods of sharp declines and then of a lot of growth. But more recently, there's generally been broad declines throughout Lake Michigan in recruitment. Now, this unit-based management is a really important part of the justification for the work that I'm going to be sharing with you today. So the idea here is that these units that are shown on the right are typically based off political boundaries and hopefully some degree of biological knowledge. And it's important to understand where the biology and these political boundaries kind of diverge. So, for instance, one of the main breaks among these is the state line between Michigan and Wisconsin. But then there's also other geographic landmarks, such as the top of the Leelanau Peninsula there, that delineates the area of WFM 06. And stock assessment that goes on in each of these units assumes that a fish harvested in a given unit originated from that unit, which is, of course, not necessarily the case. And we know that intuitively because, of course, fish don't mind invisible lines on a map. But we also have a lot of science that's helping us understand the way these fish from these different stocks or these different spawning aggregates move throughout the lake. So, for instance, this work, led by Lisa Izzo here at the Wisconsin Cooperative Fishery Research Unit, used telemetry to look at lake whitefish movements. And they tagged fish off the Garden Peninsula there in northern Lake Michigan, where the two black triangles are. And then they have a series of receivers out in the northwestern part of the lake. And here they were getting detections on the receivers across 5 different management units. So again, these fish don't necessarily stay home, and where they are harvested isn't necessarily reflective of where they came from or their stock membership. So there's a need to understand the spatial structure of lake whitefish. And again, it's important to understand the disparities between management units and the biological realities of the areas of the lake the different stocks or population segments are using, and to evaluate this unit stock assumption that there's no mixing of the stocks and that they're always just kind of staying home. So you can imagine scenarios, for instance, where maybe we're getting it right. So the colored circles I'll pop up on the map here could be the where stocks typically exist. So maybe we get it right, WFM 06 is a stock, but there's also possibility that some of these units are trying to manage or estimate population dynamic parameters in 2 different places in neighboring units that really consist mostly of a singular stock. And then there's other scenarios maybe that we can find where there's multiple stocks inside of one management unit, and we're lumping these together in our stock assessment management. So that's why it's important to understand where the biology and the management intersect. This is a really tough question to get at, and it's best achieved through multiple techniques. And there's really important inferential trade-offs in all these techniques. For instance, there's been a ton of work with physical tagging of lake whitefish that we've learned a ton from. But with physical tagging, you know where you tag the fish and you know where you capture it. So it's kind of a bookended understanding of the movement and the habitat use of that fish. And a lot of times tags are applied during spawning runs. So there's an assumption there that the fish is perfectly homing back to its natal site. And then you can think of something like telemetry, which again has been extremely informative. But there again, it's we know where we tag the fish, and then we know when the fish encountered telemetry receivers, but it's tough to understand origins. Otolith microchemistry gives us more of a chance to understand where fish came from, as long as there's enough of a difference in the chemical signature among different spawning sites. And then population genetics, which is the tool that my talk today is based on, is an exciting realm right now because of advances in technologies. They're giving us more power and more precision to be able to estimate the origins of these fish based on similarities in their genetic composition. So what I'm sharing today is not the first time somebody's thought about this or tried to do it. About 15 years ago, Justin Vandehey and colleagues here at UWSP did this for the first time using microsatellites, which was the best technology available at the time. And in this first paper in 2010, they delineated stocks among the management units and found these scenarios, as I described a few minutes ago with the colored bubbles on the map, where sometimes it seemed multiple management units contain the same stock, and in other places where one management unit contained multiple stocks. And then other times where it seems things line up pretty well, such as in the northern part of the lake with Nobbin and Epiphet up there that seem to constitute their own stock. And then this was extended into mixed stock analyses a few years later, where once the knowledge was there about what these different stocks' genetic signatures look like, then you can go out and collect fish in the open waters of the lake and assign them back to the stock they came from and start to understand stock-specific harvest rates regardless of these management units. Like I mentioned, though, there's been a change over the last decade plus in genetics shifting more toward genomics. We're now, because of technological advancements, we're able to collect data all throughout a genome, so we're not constrained to these discrete microsatellite type markers. So in that first set of studies I was talking about with the 11 microsatellite loci, you can get an understanding of what's going on there, but it's less precise. It's maybe a little bit hazier versus when you can look at thousands or millions of spots in the genome and get a much more precise understanding of what's going on out there. So there's multiple reasons for this transition to genomic scale tools, and things like increasing statistical power, like shown in the cartoon here. It gives us a chance to quantify adaptive genetic divergence. So microsatellites are assumed to be neutral, and what you're detecting with them, therefore, is basically the effects of gene flow and connectivity. But there's other important things going on, like natural selection, that can cause divergence between populations, that now we can get at these adaptive processes to try to understand them better using this genomic scale data. And finally, it just makes economic sense. Given the advancements in technology, we can get this more powerful data type for the same or sometimes even less cost than the older microsatellite-based tools. So the objectives of the work I'm going to share with you today, again, are broken down into two different areas. So first is generating foundational knowledge. And there, the first objective is to characterize genome-wide genetic structure. So for as much of the genome as we can survey, what does the genetic structure look like there? And then we want to subset just down to the loci, the spots of the genome that have evidence of being acted upon by natural selection. So we consider these to be potentially adaptive parts of the genome and seeing what the genetic structure looks like there. And then secondarily, we want to take that knowledge and I'll share some ways that we're applying it. So the first is in generating a stock assignment genotyping panel where we try to preserve the statistical power of many thousands of loci by trimming back to a very select set of a few hundred. So that way we can scale up on the number of samples that can be processed for the same price and gives this economic benefit. And then with that tool in place, updating those stock mixing rates throughout Lake Michigan was the second application I'll share. So first, we have to, again, understand what stock structure looks like out there. So with this, we have populations or spawning sites that were sampled, 17 of them from throughout Lake Michigan. There's a higher concentration of sites in the north where previously through the microsatellite work, we see there to be more genetic structure. There's more habitat variation. Reason to believe there's more genetic structure up there because of that as well. And then in the southern part of the lake, we had Muskegon samples from there. They're not shown here, but later on we did add outmigrating larvae from 4 tributaries on the western side of the Lower Peninsula, and all of those larvae were genetically indistinguishable from Muskegon. So we're confident that Muskegon is representative of other spawning sites from South Haven down in the south up through Pentwater in the north. We've checked various sites. So the way the genetic data collection worked for this is we took all fin clips from all those fish I was just talking about and extracted DNA from them. Then we used a process called rad sequencing. The way this works is we have a restriction enzyme that chops the DNA up into smaller pieces. And then those are what we end up sequencing is those little chunks, taking which gives us loci, sites in the genome from all throughout. Previous work had showed us, though, that some of those are going to be invariable. So we don't want to spend our sequencing ability or sequencing budget on spots of the genome where there is no information because everything's the same. So we used a targeted capture enrichment approach called rad capture, rapture, where we're able to target just a specific subset of loci that we know contain variation that can give us information, and that's where we gathered our genotypes from. So once we have those sequences, we call genotypes. So in the figure there, this is the type of locus this is a SNP, single nucleotide polymorphism. So it's one spot in the genome where here you have a number of A's and T's, so we have some variation. And that's the type of genetic marker used for everything I'll be sharing today is these SNPs. You'll hear me use that word a bit. So in the end, we ended up among those 17 spawning sites with 951 individuals and 197,000 SNP loci. So this is a dense data set that has information from all throughout the genome, given that, large number, nearly 200,000 SNPs we looked at. So first way we looked into these data is to just look at the overall genetic structure in the lake. And we did this in a variety of ways, but the data visualization I'm going to share with you today is a principal component analysis. I think it's a fairly intuitive way to understand the genetic relationships between these sites. So here we have a bunch of colored dots on this PCA plot, and each of the dots represents a fish. And the location on this plot is representative of how closely related those two fish are to each other. So dots closer together are fish that are more closely related. So a couple of things might jump out at you when you're looking at this first overall take at the genetic structure of all these different lake whitefish. There's some spots where we have some clustering of similar colors. So that's going to be a population or a site-specific signature that is unique. So you have some potential spatial structuring there. But the big thing, of course, is this huge split that occurs along PC1 and maybe a bit influenced by PC2, where we have two separate clouds of points. And that occurs in an otherwise nondescript place in the Upper Peninsula between Manistee and Nobbinway, where there seems to be this split in genetic structure where you have Eastern Basin and Western Basin lake whitefish. And so this was a really striking finding to us. This wasn't necessarily something that we expected. So we dug deeper into the literature to see, is there other observations that could support this pretty striking finding? And there's just another coding of it to show this east-west break. But elsewhere in the literature, the physical tagging studies that have been going on for decades, some of them were summarized by Mark Ebner in this 2010 paper. And what they found here was a really similar pattern that gave us more confidence in our finding, where the lake whitefish that were tagged to the west of this line, this red line here, in Big Bay Denok, they less than 2% were recaptured to the east. And then vice versa for fish that were tagged in Nobbinway, it was less than 3% were captured to the west. So for whatever reason, this seems to be a pretty strong break where fish just are not crossing this line that, again, it doesn't seem to be anything obvious in the lake there that causes this, but there is quite a division in the population structure at this point. So we can zoom in a bit more and look at the structure in a more regional way, given this break, this big spatial split. So first, I'll show you the eastern Lake Michigan genetic structure, which is here. So this is everything to the east and south of that red line drawn in the UP there. And what we see is pretty strong spatial genetic structure. So we've got Ingalls Point separating on that first PC axis. And then this cloud of orangish colored points at Good Harbor in Elk Rapids, which this is a scenario that's really interesting to me and I'm working to learn more about. But it's these two sites in Good Harbor and Elk Rapids that sandwich Ingalls Point that are more similar to each other than Ingalls Point, which occurs in the middle. So there's something really unique going on with that Ingalls Point spawning aggregate that makes it unique throughout the entire lake, but even among its nearby neighbors. And we also have Muskegon in the south, which is again representative of other southern spawning aggregations that's breaking out on the second PC axis. And then this cloud of points up by the straits with Nobbinway, Epiphet, Cross Village, and Little Traverse Bay. So if we zoom in on just that and run that in its own analysis, we can see a bit more structure where, again, this is kind of a gradient and a bit hazy, but we tend to find in a clustering together of Cross Village and Little Traverse Bay, so the location south of the straits, and then a separate cluster to Nobbinway and Epiphet north of the straits. So there seems to be a bit of a split there as well. And then we move to the northwestern part of the lake. So this is everything Manistee, that green MNST, and to the west. And here it gets a lot more complicated, right? This is really a big smear on a PCA. So we took this through our genetic analytical tool set and to try to understand what's going on in that part of the lake. And what we ended up finding is that there seems to be two genetic clusters occurring within that cloud of points. So this is a clustering algorithm, and the plot is showing a cross-validation error rate on the y-axis, and then k is the number of clusters it's testing on the x-axis. So the lowest cross-validation error is the most statistically supported clustering configuration. So one has pretty good support here, saying that there's just no cluster within there, but this two cluster scenario has slightly better support, and then it degrades quickly from there. So it seems if there is any clustering going on or any genetic stocks, it would be at this k equals 2. So what we did is we plotted that for all of these populations in the northwestern part of the lake and found a geographic pattern that coincided with this, where we have a northern Green Bay lineage, it seems, with Manistee, Big Bay Denok, North and Moonlight Bay, and Whitefish Bay, with the latter two being located on the lake side of the Door County Peninsula there. And then the southern Green Bay, we get a much greater prevalence of the lineage shown, indicated here by the blue, where in Menominee, Peshko River, Larson's Reef, Riley's Bay, and the Fox River, more of this southern lineage, but still obviously plenty of mixing between the two. But based on this and the geographic clustering of these, we feel like there's likely two different lineages that are spatially overlapping, perhaps, there in the northwestern part of the lake. So what we want to do is then take, instead of looking at all 197,000 loci, and we want to thin that down to just the ones with signals of adaptation and look at how those are structured throughout the lake. The concept here to be able to choose or identify which loci have signals of influence by selection, which might be adaptive, is that the loci with greater divergence than is expected suggests that selection is occurring. So FST is a metric in population genetics that it summarizes the genetic difference between sets of samples. And this can be calculated throughout a whole genome or at individual spots in the genome. So you can imagine a scenario where across an entire genome, we have an FST value of 0.2, which indicates some modest level of differences between fish throughout the genome. But then you might find a given genomic position that has a greater FST value of 0.8. So that suggests that something is driving that part of the genome to be different. So it could be a part of the genome that codes for some trait that's under selection. So this is the general logic behind how we're identifying adaptive loci. And we did this process using a program called PC Adapt, which all they need to know about that is it's robust to add mixed individuals, which we have many of, and it also handles hierarchical population structure well, which again we have evidence of. These results can be shown in this sort of a figure. So here on the y-axis, that's a measure of statistical significance. So as you go up on the y-axis, you have more evidence that a locus is under selection. And then along the x-axis, all of those numbers are chromosomes. Each one of the dots is one of these 197,000 SNPs that we looked at. So if we take all of our populations together and run this analysis, we find that there's quite a few spots where there's outlier loci. One of the things that we like to do to make sure that we are highly confident that this signal is real and not a statistical artifact is to look for loci that are bunched up physically close together on chromosomes. When that happens, it's because they're in physical linkage and it's a stronger signal of selection. Those scenarios are shown with these purple bars that are vertical. So on chromosome 4 and chromosome 11, we have a pretty strong signal that there's selection occurring at those spots on the genome. But then we also, because we have this hierarchical spatial population structure, we wanted to look at just different parts of the lake. So here, this figure is the same sort of thing, but just looking at the spawning sites in the northwestern part of the lake. And for this, we have this part of chromosome 20, where there's this big cluster of loci that are all behaving similarly and as they're looking like they're under selection. In the east, we have 3 chromosomes 7, 10, and 18. And in the northeast, this one on chromosome 10. So it seems to be these regional patterns of selection going on. A way that this can be visualized more precisely is to look at these spots on the different chromosomes with signals of selection and see how they vary among individual sites. So this figure here shows for that group of outlying loci, the group of loci under selection on chromosome 7, the shading indicates just how genetically different that part of the chromosome is among the different sites. So as we get more up this red scale, it indicates a greater genetic divergence. All the X's on here indicate non-significant differences. So on chromosome 7, a quick takeaway from this figure is that it seems to be Muskegon that is quite different from the rest of the populations at this point on chromosome 11 or 7. So could be something going on there with the Muskegon group causing selection at this site. Just briefly overview of the other chromosomes where we saw these strong signals of selection. On chromosome 4, we have differences going on there for Cross Village and Little Traverse Bay, as well as that Ingalls Point site. This was the same chromosome 7 one with Muskegon. Again, chromosome 10 is pulling out Cross Village and Little Traverse Bay mostly. Elk Rapids and Ingalls Point on 11. Same, Elk Rapids and Ingalls Point on 18. And then a weaker signal, but still a consistent one, of Fox River being differentiated at that spot on chromosome 20. So it seems to be these, a lot of times what we test for is outlier loci like this that are in selection throughout an entire system. In this case, it seems to be really these localized forces that are leading the way. So a logical extension of this is, what might be causing this selection at these places. So for that, we performed a redundancy analysis, which looks at covariation between allele frequencies and environmental variables. We looked at 7 environmental variables, all collected via remotely sensed data, and we did this at both a local and regional scale. So local is just within 5 kilometers of a spawning site, and regional is 50 kilometers. Looked at cumulative degree days, percent sandy substrate, shoreline sinuosity, which is just how much variation there is in shoreline contours, surface temperature, percent annual ice cover, spring warming rate, and depth. At the regional level, a few of these were collinear with other variables, which breaks an assumption of the analysis. So the ones that marked as collinear did not go into the analysis. But again, this analysis lets us look at whether there's relationships between the genetic changes and these environmental factors. And we've corrected for multiple testing of all these different loci using a false discovery rate approach, which is standard in population genetics. What we found is each one of the orange dots there is a locus that was an outlier and related to some sort of an environmental variable. And then the biplot here shows each of the arrows. The longer the arrow, the more influence it seemed to have on these different genomic areas. And the big takeaway here is that a lot of these all have local, that have the strongest signature. There were a couple significant ones, regional, but less important. So what it indicates to me is that these environmental selective pressures are likely operating at the scale of the very localized spawning site and could be influencing selection through factors associated with spawning and reproduction. So with that knowledge, we wanted to be able to start applying it and help inform management. One way is to try to come up with a genotyping panel that captures the information I've shared so far statistically, but just in a smaller number of loci. And to do this, we first need to aggregate these spawning sites into stocks. And this is what we're referring to stocks, or referring to as stocks or reporting units. And this is because not all of these spawning sites are genetically distinguishable. Some of them just look extremely similar, so we can lump them together. And we use a lumping scheme based on the observed genetic structure, like what I was showing on the PCAs, and also shown that we got through other analyses that I didn't show in this talk. So the goal of this genotyping panel design is again to capture the statistical power of genome-wide data using a smaller number of loci. And the point of that is to reduce genotyping costs so we can scale up our data collection and ask bigger questions. The way we did this is by selecting loci that had the highest FST among the delineated stocks, so which are the most informative for splitting out these stocks in the way that we saw them to be split out with the 197,000 loci. And the type of panel we designed is a GT-seq panel, which is basically highly multiplexed PCR. This is the way we lump things using the management units as a unit here as well. So what we have is this GMZ1, so genetic management zone 1 in the northwestern part of the lake with northern and southern stocks that are colored the same here in green. GMZ2 in the northeastern part of the lake, GMZ3 based on the Good Harbor and Elk Rapids and East Grand Traverse Bay signature, GMZ4, which is in West Grand Traverse Bay, because that Ingalls Point population or spawning site is just so different. We feel like it needs to be accounted for individually. And then GMZ5 points to the south in Lake Michigan. Our genotyping panel design went well. So we started with about 1,400 candidate markers that had these high FST values. And then through a series of quality filtering approaches and making sure that we could robustly amplify them using this GT-seq technique, we got down to a set of 472 that did a good job capturing the genome-wide variation. And we can assess the accuracy of this tool by looking at reassignment rates. So if we sampled a fish from a given site, and what's our ability with this tool to be able to reassign it back to the site we know it came from? And we do pretty good. The one spot where it gets a little iffy, the 76% reassignment rate, is GMZ3, which is this Good Harbor Elk Rapids one. GMZ or WFM06 seems to kind of be a crossroads of the lake where there's a lot of different things going on for various stocks. So that's why it gets a bit noisier. But elsewhere, we had 91, 97, 99, and 100% reassignment accuracy. So feel like the tool is doing its job. And so we want to take that tool and apply it to look at stock mixing rates throughout Lake Michigan to better understand if you go out to the lake during a non-spawning time and you take a sample of lake whitefish, which stocks are contributing to that area of the lake at that time is what we wanted to ask. So for this, we use that 472 SNP GT-Seq panel, and we had 44 sample sets collected from 1977 to 2022, so a total of about 3,500 samples, with that big sample size being possible because of the use of this GT-Seq panel. This is the way those sites were distributed through time and across the management units. So some going back to 1977 in Wisconsin, for instance, as well as the 80s. Then we had more collections throughout after the turn of the century in the 2000s and the teens. And then 2022 was when we did sampling for this study specifically, where we wanted to look at seasonal variation in mixing rates. So we collected samples in the spring, summer, and fall. This is what we learned in doing that. So in WI2, which is mostly on the lake side of the Door County Peninsula, but then wraps around a bit into Green Bay, we looked at seasonal variation. So between summer and fall in 2022, in the summertime, we had quite a bit of contributions in the site from both northern Green Bay and southern Green Bay. But then in the fall, it shifted more to a northern Green Bay dominant composition, which makes sense because this is some of the spawning locations that constitute that GMZ 1 North lineage. And so what they're doing there is coming back home to spawn in the fall. Saw a similar pattern here in WFMO2, where during the springtime, we had contributions coming from the neighboring area to the east. But then in the fall, it was the more local GMZ1 North stock that made up the entire catch. Looking at short-term interannual variation, as well as some of the seasonal differences, here in southern Green Bay, we're seeing similarities between 2020 and 2022. In the fall, though, we get the same pattern where the local, that GMZ1 South stock tends to be coming home to spawn, so we see an increase in prevalence of those fish. And over in WFMO6, that's what I mentioned a minute ago, it's kind of the crossroads of the lake is what I think of it as. And we're getting contributions from all the different stocks, except for that southern Green Bay stock here. So, and that was a similar signature between both summer 2020 and summer 2022. Is just a lot of contributions from a lot of different stocks going on in this more central area of the lake. Looking at longer term patterns, so here we're looking from 1997 to 2022 in the southern part of the lake. For a while, it was simply the local stock that was getting seen in this area. But then more recently, maybe some contributions from that GMZ1 North stock and GMZ2 in the northern part of the lake, some fish that might be wandering down during the non-spawning season. In the northeastern part of the lake, this is kind of an interesting story where in 1977, 86, 93, catches were purely comprised of that GMZ1 North stock to the west. But the more recent snapshot in 2022, it had flipped to being the local stock instead. So that might indicate maybe some increasing prevalence of that stock, more of those fish out there that are showing up in the 2022 sample. So some of the take homes from this work I've shared with you today is that there's this major break between eastern and western basin lake whitefish. And then in the east, we have this site-wise, spawning site-wise spatial genetic structure that's evident. And in the northwestern part of the lake, these spatially overlapping lineages. Adaptation seems to be site or region specific, and it looks like some local environmental factors may be important for driving some of those adaptive processes. For the mixed stock analyses, looking at seasonal variation, what we picked up on, we think a lot is spawning migrations as more local stocks become more prevalent in the fall. Across short-term interannual variation, and as well as long term, it really depends on the region of the lake. Sometimes it's pretty consistent across years or even decades, but in others, we saw some pretty big swings, like in the northeastern part of the lake, where the local stocks seem to kind of have a resurgence, maybe. The GMZ1 North in northern Green Bay, we saw that stock in just about every mixed stock sample that we analyzed. So it seems to be most ubiquitous and kind of providing a lot of the whitefish that are getting captured throughout the lake, at least to some degree. And then we're really happy with the performance of the GT-Seek panel, so it's enabled accurate and large-scale stock assessment. And I just wanted to quickly, kind of maybe the lightning round of the talk here, share some other things that are going on, exciting projects with lake whitefish genetics and eDNA research all throughout the Great Lakes. So one thing that I'm working on is doing some whole genome sequencing to better understand the genetic breaks I was talking about in this data set. So I'll be resequencing some of those fish throughout their entire genomes to better understand patterns of adaptation and maybe get a better idea of what makes Ingalls Point so unique, for instance. In Green Bay, leading a project supported by Great Lakes Fishery Trust, we're using eDNA surveys to try to find reef spawning locations for lake whitefish in the fall, and that's in kind of a pilot phase right now, but is going quite well. In this WFM06 area, working with Ben Marcy Quay from Hammond Bay and others on a Great Lakes Fishery Trust funded project to use close kin mark recapture to try to estimate stock sizes through time using historic samples in that part of the lake to see whether this close kin mark recapture framework could be a way forward for abundance estimation. On Lake Erie, Great Lakes Fish and Wildlife Restoration Act is supporting a project led by Peter Euclid at Purdue that I'm helping out with. We're using whole genome sequencing mostly to try to look at stock structure in Lake Erie, where the GT-Seek panel indicated more subtle patterns. I mentioned the environmental drivers, the local adaptation work that I'm working on in Lake Michigan. So more to come on that as that paper comes together. Some exciting stuff funded by one project by the Great Lakes Fish and Wildlife Restoration Act and another by Commission is combining inferences from genetics and telemetry in northern Lake Michigan as well as Lake Huron. So like I mentioned, these tools kind of have different strengths. So if we can use their inferences in a complementary way, I think we can paint the most complete picture. So working on a couple different projects there. And then working with Amanda Akis, the Great Lakes Science Center, and Chris Wilson, they're in Ontario, to try to pull together more of a whole Great Lakes understanding of stock structure, maybe at a coarser scale within each of the lakes, but trying to understand on among each of the lakes, what does stock structure look like for the lake whitefish. And so I just want to conclude by thanking the sample collectors for all of this research. A ton of work went into getting these, and especially Commission and Trust for funding the bulk of the research I shared with you today. And I'm happy to answer questions now, and also happy to chat with people via email as other things might come up. Thank you.
Fantastic. Thank you so much, Jared. I like how you ended it there by talking about sort of next steps and sort of cross-lake studies as well. That's really encouraging to hear. I hope we are going to get a lot of questions. People can offer their questions up via the chat, or preferably you could just throw your camera on, raise your hand, throw your camera on, so we can make it a little bit more personal. Before we go into the questions, one thing I forgot to mention is that we do have a tentative speaker for December. His name is Colin Bean, and he is going to be talking hopefully about some of the restoration strategies they've used in the United Kingdom for some of the European whitefish and Vendace species. So look for an email from Jake about that. But we also are very much interested in recruiting some other talks. So feel free to reach out to Jake or myself if you'd like to give a presentation to this very friendly and friendly group. So, okay, I know we've got a lot of geneticists that I hope are going to ask you some questions. So I'm going to come at you from a non-geneticist perspective, Jared. Um, Really amazing to see that east-west split, as you said, between Manistee and Nobbin Way, and then even more so that sort of Ingalls Point showing up differently. And I wonder, I'm sure you've talked to some of the fishers or some of the biologists around there that are working sort of those areas. Is there anything that sort of jumps out functionally or morphologically? Let's say, let's just stick with the Ingalls Point stock that seems to suggest why it might also have this unique genetic sort of difference.
Nothing that I've been able to get from anybody. It's a lot of head scratching going on right now
Huh.
about what could be driving that. I'm not aware of any morphological differences or behavioral differences or spawn timing or anything like that. And especially the Ingalls Point one just seems so unique because it's
Yeah.
so different between East and West Grand Traverse Bay. I mean, you can stand in Old Mission Peninsula and see both those sites, right? They're so close together.
Yeah.
So I unfortunately don't have much of a lead on that, and I would love to hear from the group if anybody had any
Yeah.
observations or thoughts on what could be driving either one of those, really stark patterns in these data. Oh, you might
Okay.
have some ideas.
Here we go. Yeah. Chuck, I saw your hand first.
Yeah, great talk. And I love how you kind of opened the black box for us non-geneticists and kind of explain things. That's very helpful. So, I run a coded wire tag and tag recovery program, and you know, we see of the fish. Now we're looking at stocked fish, um, Chinook, steelhead, and lake trout. And it seems like the ones that get tagged, stocked and tagged in that Grand Traverse Bay area have the most restricted movement when we look at the recapture information. So it may not be genetic, it may be something in the physical environment. It's very strange that we see these kind of things. When we look at recoveries of fish that were stocked elsewhere, I mean, there are more and more, they're all over the place. But the fish stocked there are just, and they don't move much. And, and that's a funny area because the steep drop off between those two basins, it's very sharp. And you go I mean, you go set some gill nets there, and boy, you're in 400 feet of water and about 8 corks or something. It's amazing. So it's very strange. But that's all I have to contribute there.
Yeah, thanks, Chuck. I appreciate that. That's interesting to hear the similarities between the species like that, and that's yeah something we ought to think about together a bit.
So maybe just to build on what Chuck said, Chuck, so you're suggesting they're really maybe not moving much from Ingles Point, but they're still homing, and what Jared could be picking up on would be just drift through time because of sort of that reproductive isolation because of the physical habitat.
You asking Jared or I?
Either one. Maybe.
Yeah, I think there's something in the physical environment that's,
Yeah.
yeah, separating them or whatever. Yeah.
Yeah, and it could certainly be both if there's some differences in habitat that maybe there's a selective thing there going on where maybe it's, different substrates or different currents or that sort of thing. But then like you're saying, Chuck, just the environment keeping them from mixing, it could be helping to kind of pour fuel on the fire of any selection that's going on as well. So it could very well be both.
Okay. Thank you. I have a
Thanks, Chuck.
question, but I'll let the others go ahead. Yeah.
Okay. Yeah. Yeah, let's go to Ted, and then Wendy dropped something in the chat, and I'll go to her next to have her maybe explain that figure. But yeah, Ted.
Thanks, Jared. It's always fun to see updated results of all this. I had three questions, but I think I went over them down to two. So just something that jumped out at me, like the 0405 units looked like it only had samples from a time, maybe, I don't know, 20 or 30 years ago. I can't remember exactly what it was. Just wondering, and then when you showed three, it showed, it looked like it was distinctively, I think, the northwestern unit, and then became distinctly something else. I think the unit three. Wondering if there's any chance that maybe all those could be explained by some influx of fish from Huron. Maybe that there something happened and they either died out, left, whatever, and it was recolonized by fish coming from Huron. I'm not sure if there's any genetic samples you could use from the other side of the straits. Um, and then the other question was, I was just thinking about kind of what the Dans from Stevens Point and saw in the telemetry stuff in terms of there's a lot of whitefish that spawned in, say, the Peshtigo, the Menominee, those rivers up Green Bay, and then as soon as they got done spawning, they hightailed it down to the Fox River. It wasn't a ton of them, but I'm wondering if that could lead to a bunch of kind of the mixed signal in Green Bay, and if there's any way you can tie those fish to. I mean, maybe the sample would only be 5 fish where you have genetics and good data where for a number of years after what they spawn, but it'd be interesting to find out if that stock mixing is real stock mixing or if they're actually spawning where they are and then they just get harvested or caught somewhere further away just because that's what they do every year. So really great stuff though.
Thanks, Ted. And yeah, I appreciate your contributions to these projects as well. So on the first question with Lake Huron inputs, fascinating idea. Haven't looked into it explicitly, but we're getting more and more data from Lake Huron with projects that are underway right now. That's going to make that much more possible as we move ahead. So it could explain that. But I suppose stay tuned. That's within a couple of years, we should be able to give that question a thorough treatment. And then, yeah, the stuff in Green Bay, that the genetics and telemetry project that I mentioned going on that part of the lake, that's exactly those fish, going back to some of the existing telemetry data from years past, and now genotyping them and assigning them to stocks, which I actually later today need to send those results to Demkowski to tie our stock assignments to the telemetry tag numbers. So yeah, that to be determined there as well. I think there's a lot of different factors that can be influencing that Green Bay signal, from population expansion that we've seen, maybe some recolonization dynamics, and then, like you say, they seem to be pretty fluid in how they move around and use that system. So it certainly seems distinct from the way the rest of the lake is structured based on the work we've done. But yeah, the telemetry plus genetics is going to be the coolest part of that.
Cool. Thanks.
Thank you.
Thank you, Ted. Wendy, did you want to explain what you dropped in the chat next?
Yeah, it just sort of that striking sort of divergence area just kind of popped into my head that's sort of a location of the Lake Michigan. It's called the Michigan Basin Sill, which kind of marks an uplifting level that marks sort of a low water of late Lake Chippewa. So we're talking back in the geological history that was a level that was an area that marked changes in water levels that might have at some point separated some of these groups of fish. So you might be seeing sort of a signature of past separation there.
Yeah.
It's just kind of funny if you drop sort of like the mapping, some of those maps that you showed of where the splits are on some of those features, they line up pretty nicely.
That's awesome. Thanks for pointing that out, Wendy. I might follow up with you via email to chat more and get the figure and source for me on that. That's great.
Yeah, very cool. Thanks, Wendy. Jory.
Yes, hi. I have an anecdote and a comment, which I'm sure they're engaged with you, but I just want to check. So it would be interesting to look at Hondorp's movement which I don't focus on the whitefish side of that as much, although I don't think they're moving as much as our Cisco are. But if you have that Lake Huron story, is that showing up in Hondort's movement studies or anything like that? Then my anecdote is with lake trout, and there definitely is in that bay an odd separation and a reluctance to cross the deep water. And my anecdote is stocking lake trout. A number of years ago, I moved everything over to East Bay because the habitat is better for lake trout, never considering that they wouldn't swim across the bay. And the fishery completely tanked in West Bay. And I started getting phone calls about, 5 to 7 years in, when you would expect to see them show up in the fishery, and they don't have any fish over there. And so I've had to reallocate the stocking because of that reluctance to cross the bay. And it seems like such a small thing and it wouldn't be an issue, but it really was a big issue. And so it sort of feels like I'm hearing that same story here, that you have these units that are just not wanting to go across for whatever reason. And then why the right side is related to MM5 is intriguing also. And it would be interesting to look at bathymetry there, or, what's keeping them in.
Yeah,
Yeah.
I agree. It seems like they're able to kind of go from East Bay out and around the Horn, maybe, but
Yeah.
not so
Why?
much straight across. So yeah, I appreciate that, and I'm working with Gerald and other folks on that work, so we'll be able to put some stock assignment information to that as well to see what we can learn. So, thank you.
Yeah, thanks, Joy. Yeah, Chuck.
Yeah. So my question, and thanks, Jory, for that. That's great stuff. My question is given what you showed us, the this idea of homing and the specific natal areas and stuff, it looks, like there's some of it, but it looks pretty soft. I mean, it's more regional, and that certainly kind of gels with my conceptual model of Coregonines. And it's not like salmon or certainly sturgeon or much older uh, fishes that have gone through, very well and developed well stock isolating mechanisms. And I assume this data is supporting a much softer homing ability. I think strain is adaptive and important. It's not a bad thing. So anyway, what's your thoughts?
Yeah, I agree. A couple thoughts on that. So overall, the shank structure we saw here is pretty subtle. Like these FST values are pretty low across all these different areas. So there's certainly a gene flow part of the story where there's a lot of mixing going on at spawning time. Well, when I say a lot, there's a non-trivial amount to keep the genome-wide differentiation low enough. But on your point, use the word regional, which I think is perfect for this. And it's one of the places where I'm excited about combining what we're doing here with the telemetry is it seems like there might be phylopatry to a region of the lake or maybe a set of tributaries or reefs.
Yeah.
So what it seems genetics can do for us is get us in the right neighborhood, but then we really need to use something like telemetry to understand, what's the relationships between the houses and the neighborhood, for instance, to use that analogy. Like, I think it's a place where we're going to find a lot of strengths where you use multiple tools to tell the whole story. But these data suggest to me that it is more of a regional rather than a really site-specific thing, with the exception of maybe like that Ingalls Point site that seems so different.
Right. That's the exception rather than the rule. Yeah.
Seems to be.
Okay. Thank you very much.
Yeah, thank you.
Yeah, Amanda.
Hey, Jared, nice talk. I enjoyed listening to it. So I was particularly intrigued by one of the last slides you showed, where there had been sampling over time in the Nobbinway, like Northern Shore area there, where you had seen this sort of regime shift. And I was looking at the dates and also looking at the dates of the individuals that were used for the rapture analysis where the panel was developed. And those were caught, because you mentioned 2022, you're like, it's more local fish. But I'm like, are they local, or is there some kind of shifting of that line there happening over time? Because the fish that were rapture sequenced and that the GTC panel was developed off were collected in 2019. And I was intrigued too by in your PCA where there are a few things that are East fish that are over there. And so I was curious your thoughts on whether or not that actually might be a little bit reflective of like population demographics sort of shrinking on the Green Bay side of things, where you might have had sort of spillover from that habitat all in Green Bay that would be, you know, ideally great spawning habitat. And with all of the, you know, things that whitefish has been going over, might you be seeing sort of a shift in where that stock line is occurring over time because of some of the impacts on these populations?
Yeah, I don't have necessarily a lot to offer beyond what you just explained, Amanda. Like that sounds reasonable to me. It could be sort of a moving of where that split's occurring and the areas these stocks are occupying, as reflective as anything. So, yeah, I don't have much to add. It seems like a reasonable explanation, though.
Okay. Do you have any sort of plans to do any sort of demographic analysis with some of the data that you have or are generating in the future? That's just sort of like a personal curiosity, since we're, I think, going to be starting some of that as well in Lake Michigan for Cisco. And I also will point out, we did specifically, having seen the results that you guys got in Ingalls Point, we did look at Cisco that were caught in Ingalls Point, and they do not show any kind of view. They're definitely Elk Rapids, Grand Traverse.
Oh, wow. Nice.
Classic. They don't show that same unique pattern that the whitefish show there. So I'll be curious to see if Jory's hypothesis about the deep water being a bigger barrier for whitefish might be at play. But
no, that's super interesting. Yeah, and I have another more than 100 Ingalls Point fish I need to run yet to help make sure this wasn't kind of a fluke for whatever reason. So yeah, much more to go on there, but no demographic analyses teed up. But it sounds like something this could be ripe for, and especially if you all are kind of thinking that way already and for other species. Maybe we should talk more and dovetail a bit.
Absolutely.
Great.
All right. Thanks, Amanda. Wendy.
Hi, so thanks again, Jared. This was great. I was kind of interested in some of the habitat, like relationships between the genetics and some of the environmental variables, particularly some of the habitat and the shoreline sinuosity, because I've been thinking about that in like whitefish from Lake Huron from some work I'm doing, and sort of did think about it in some Cisco work. And I'm also thinking about it for some Arctic salvelinus work where we have, like a lot of salmonids, we have some species that like to spawn on piles of rocks and some that spawn in the same space but don't spawn on the piles of rocks, and just sort of thinking about how to measure those variables, because I see when you were talking about some of them, they come up collinear with other variables, and I maybe this isn't a question just for you, but maybe for other folks that work more on habitat, just how to get good measures that capture that preference, and sort of looking at structure where you have like all these nice shoreline nooks and crannies versus places where you don't, and just how to get a good way to map and measure that is something I've been struggling with a little bit.
Yeah, I've had those same thoughts, but yeah, I wish we had finer resolution data, because like you're saying, it could be like a really hyper local thing that's influencing this, and we're kind of restricted by the remotely sensed data that we have, or data drawn from models. So I feel like that is kind of a shortfall of this, is we can get fairly local, but it's some of it's based on interpolation, and I don't think we can get to the level that's most meaningful to the fish a lot of times. But if, again, like you said, Wendy, if there's other folks on the call that have more experience in the habitat realm, I'd love to hear possible solutions.
Yeah, thank you, Wendy. Good question. Good thought. And I think also like showing the, obviously very different in terms of like shore sinuosity, the north and the south, other environmental variables too, like probably ice cover, which probably has led to way more in climate, way more whitefish to the north. But I mean, the south, it's didn't find much there, and it seems to be one sort of Muskegon or larger stock, and of course their densities aren't as high either. So Jared, one question as you got into some of the adaptive chromosomal work, I thought that was really interesting. And for example, you had like the Fox River, it was different than all the other ones for one chromosome, I think. And it got me thinking about sort of moving towards like what, and I don't know how you all do this. Trevor Kravinoff has done this some, but how do you move to the point of like saying, okay, what is actually coding on that gene that has this higher FST level? Is that something you can do by just looking for other sort of salmonid sort of functional things, or you didn't mention that in sort of future work, but I wondered if you were interested in that or just didn't mention it.
Yeah, I'm certainly interested in it, but some of the trying to assign specific traits to the genomic positions, like it's really kind of a sticky area that's based on a lot of correlations, and I'm specifically a bit weary of extrapolating too much on that. So, I think there's more that can be done on that, but I'm personally a little bit gun shy to tie specific traits to it based on genome annotations, for instance.
Yeah. Okay. Oh, fair point.
Yeah, I mean, some of my other genetics colleagues on the call might have more nuanced answers than that, but that's where my mind is currently.
Yeah, Trevor was on earlier, but I don't think he's on anymore. So, Wendy. Was that an old hand, Wendy, or did you have another question?
Can you hear me now?
Yeah, we got you.
Okay. Yeah, just another question just popped in my head. Sort of, you were talking a little bit about some of the ongoing work on Lake Erie, and one thing that kind of interested me there, in light of some work I've been doing on the Detroit River, was that some of the structure you saw was basically that they did see was driven in large part by an inversion, that in an area that in other species is sort of associated with migratory capacity. I was kind of interested in some of these differences you were seeing where fish were moving more often. Did that same inversion come into play at all?
The inversion in the sheet paper, or which one is it, Wendy?
Yeah, it was the Schmidt paper.
Oh, I'm not sure. I see I see Peter's name. I don't want to put you on the spot, Peter, but do you have any insights in that?
Yeah. Um, yeah, I don't know. So that was with the same panel. That was Rapture data. And it did look like it had an inversion. And it's actually, I think, in a similar region as some of the stuff that Jared presented today. Um, so I wrote about it in that paper and still talk about it as a very putative inversion because it can be hard to identify them consistently with short read data like we have. But the whole genome sequencing stuff that Jared's talking about in Lake Michigan, and then we'll also have that whole genome sequencing data for Lake Erie. And I'm hopeful that in both of those data sets, we might have the high enough resolution to sort of answer that a little bit more fully. Because Because yeah, I think that inversions are kind of one place that can harbor genetic variability in these high gene flow populations. So I think there's a lot of potential for them to be involved in these sort of slight genetic differentiations that we see in these islands of divergence, but they're just so hard to conclusively identify without Trevor helping you out and getting long read data that can cover the whole region. So that would be, next step is the whole genome sequencing that Jared and I talked about. Step after that would be getting Trevor involved to sort of conclusively identify exactly the endpoints of any inversions that we'd find.
Great. Thanks, Peter.
Yeah. Way to put him on the spot and wait for Peter to jump in there. Like
Nailed it.
Yeah. All right. Any final questions? We've still got almost 30 people hanging on listening, so that's great. If not, give the virtual round of applause or whatever, way that you can do that to congratulate Jared on a good talk. He said, feel free to reach out to him via email, and good luck with the move, Jared. And we're glad that you're staying in the basin and continue to work on these issues.
Great.
So,
Thanks,
all right.
folks.
Thanks, everybody. Have a great weekend.